Hi everyone, I am Dr. M. Parimala, Assistant Professor, PG and Research Department of Microbiology, St. Joseph's College of Arts and Science, Autonomous, Cuddalore, Tamil Nadu. I am going to present a topic gel exclusion chromatography. It is otherwise called as gel permeation chromatography. Now let's see the principle of this gel exclusion or gel permeation chromatography. It is the technique in which the separation of the components, that is from a mixture, the analyte can be separated, and it is based on the difference in their molecular weight or molecular size. The stationary phase is used in this is it is the porous polymer matrix whose pores are completely filled with a solvent to be used as the mobile phase. It involves a stationary phase as well as a mobile phase. The molecule which is present in the sample are pumped through the specialized columns that contains such microporous packing material. That is, the packing material is the gel, which is the solid, that is stationary phase. The basis of the separation is that molecules above a certain size are totally excluded from the pores, while only the smaller molecules can pass, that is access into the interior of the pores, that is the pores of the gel, either partially or wholly. The flow of the mobile phase hence will cause larger molecules to pass through the column unhindered. So while passing through the stationary phase, the larger molecules cannot penetrate into the gel. So it will move through the column without any hindrance. Whereas the smaller molecules will be retarded according to their penetration of the gel. So based on the size, the molecules get hindered or it can move very freely through the column. That is the stationary phase. And now let's see the components or the instrumentation of the gel permeation chromatography. So it involves the usage of a stationary phase, then the use of a mobile phase, then the use of different types of columns are there, then there is the pump for passing the sample, then detectors are there, then we will see the things one by one. First one, stationary phase. Stationary phase is composed of semi-permeable porous polymer gel beads with a well-defined range of pore sizes. So we have already discussed this. There is the stationary phase in which we are using a gel. The different materials of gel beads are available with definite pore size. So through this only that is through the pores the molecules can pass through. Only the smaller molecules can pass through, while the large molecules will be excluded. And it has the following properties. Since it is the stationary phase, it should be chemically inert. That means it should not react with the analyte. Then it should be mechanically stable. Then it is ideal and with homogeneous porous structure, that is pore size for better resolution. Then a uniform particle and pore size. Then we will see about the different types of packing materials, that is the gel that is used in this gel permeation chromatography. First one is the dextran. It is Cephadex gel. Actually, it is the 1,6 polymer of glucose natural gel. Then agarose gel. It is a 1,3 linked beta D galactose and 1,4 linked 3,6 anhydro and L-lactose natural gel. Then acrylamide gel can be used. It is a polymerized acrylamide, a synthetic gel. So these are the different types of gel materials that is used. And then the mobile phase, it is composed of a liquid used to dissolve the biomolecules to make the mobile phase permit high detection response and wet the packing surface. Then the columns which is preferable for carrying out this chromatography is analytical column, which is 7.5 to 8 mm in diameter. Then preparative column 22 to 25 mm in diameter. then the usual column length will be 25, 30, 50 or 60 centimeters. Narrow bore columns can be used that will be 2 to 3 millimeter in diameter. Then pumps. The pumps are either syringe pumps or reciprocating pumps with a high constant flow rate. Then detectors. The detectors may be concentration sensitive detectors with bulk property detectors or refractive RI detectors, etc. So these are the different types of detectors which can be used for this chromatographic technique. In this picture, you can see very clearly how this gel permeation chromatography it works. It consists of a column. The column contains the packing material, that is the gel material that will be arranged inside the column, and through the mobile phase, the sample should be injected that contains the different sizes of molecules are there which will pass through the stationary phase, that is the gel with a definite pore size. So if the molecules, the analyte, if it is found to be smaller, it can pass either partially or fully through the gel, while the larger molecules are excluded and it will be passing directly in between the gels. So in this, you can see in this A column, there is the sample injection. Then in the B column, the sample separation is occurring. Then in the C column, since the larger molecules, they will be they will not be entering through the gel pores, so it will be easily moving and that will be excluded first. Where the smaller molecules, since it moves through the porous structures of the gel, it will be moving slowly. So based on the retention time, the different molecules get extruded out. We will see the steps. It involves three major steps, that is for running this chromatography. First one, preparation of column for gel filtration. So first one, it involves the swelling of gel. So whatever the gel what we are using for this preparation, we have to swell the gel. Then pack the column semi-permeable porous polymer gel beads with well-defined pore size. Then afterwards, washing step is that after packing, several column volumes of buffer solution is passed through the column to remove any air bubbles and to test the column homogeneity. So there should not be any air bubbles. Then we have to load the sample into the column using a syringe. Then eluting the sample and we have to detect the components. And now let's see the applications of this gel permeation chromatography or it involves this method is mainly exclusively used for the fractionation of the proteins as well as the purification of proteins, and also it is used to find out the molecular weight of those compounds, separation of sugar molecules, proteins, peptides and others on the basis of this size, and it can be used to determine the quaternary structure of the purified protein. Thank you.